After completion of human excretory system, now we want to focus on the details of nephron or uriniferous tubules. You know that what is nephron or uriniferous tubule? Nephron or uriniferous tubule is the structural and functional unit of kidney or excretory system. Each kidney consists about 1.2 million nephrons or uriniferous tubules. Nephrons are the microscopic filament-like structures. Average length it is 3 to 6 centimeter, while the average diameter it is about 30 to 40 micrometer is the average diameter. Now concentrate on this microscopic structure of nephron or uriniferous tubules. This nephron or uriniferous tubule consists long tube-like structure which is blind at anterior side while remains open at posterior side, and it differentiated into different part, that is the Bowman's capsule, PCT, proximal convoluted tubule, then loop of Henle, distal convoluted tubule, and common collecting duct. So this is the long tube consists different component, that is first Bowman's capsule, neck, proximal convoluted tubules, loop of Henle, distal convoluted tubules, and terminal duct. So it is a long tube. At anterior blind portion which form the cup shaped structure in which glomerulus associate, and glomerulus and Bowman's capsule together forms the most important structure, that is termed as a Malpighian corpuscles, or it is termed as a Malpighian body. This Malpighian body discovered by the scientist Malpighi, so it is termed as a Malpighian body. Now concentrate on this Malpighian body or Malpighian corpuscles. It is first component of this Malpighian corpuscles, it is cup shaped cavity. It is termed as a Bowman's capsule. So this is the cup shaped cavity termed as a Bowman's capsule. It is discovered by the scientist Bowman, so it is termed as a Bowman's capsule. This cup-shaped Bowman's capsule is made up by two layers. This is outer layer is termed as a parietal layer, while inner layer is termed as visceral layer. So this Bowman's capsule made up by two layers, that is outer parietal layer and inner visceral layer. This outer parietal layer is lined by the flat squamous epithelial cells provide the protection, but visceral layers lined by specialized squamous epithelium, and this specialized squamous epithelial cells called the special name, it is termed as a podocytes. So it lines by special cells termed as a podocytes. So here it is presence of the podocytes like this. So this is the podocyte. When we focus on the simple structure of podocyte, these podocytes have the foot processes like this. It have the foot processes and microscopic slits are associated. So peculiarity of podocytes have the foot processes and that is slits, or it is pores. So this is the podocyte. So what is the podocyte? Podocytes are these specialized squamous epithelial cells which lines the inner layer of Bowman's capsule termed as a visceral layer, and this is plays important role in ultrafiltration. This plays important role in the process of urine formation. It is termed as a ultrafiltration. Now the space enclosed by these two layers, parietal layer and visceral layer, it is termed as a capsular space. It is termed as a capsular space, also termed as a urinary space. Capsular space or urinary space. So capsular space lies between the parietal layer and visceral layer. So this Bowman's capsule have average diameter, it is 200 micron is the diameter. So largest diameter of the renal tubule observed in the Bowman's capsule. This Bowman's capsule have formed a cup shaped struck cavity, cup-shaped cavity, in which the mass of blood capillary is situated, which is arises from the afferent arteriole, and it leave the Bowman's capsule as a efferent arteriole. So this is termed as a afferent arteriole. It is termed as afferent arteriole. This afferent arteriole, when enters into the Bowman's capsule, it forms the tuft of capillaries, mass of capillaries. So here it is formation of tuft or mass of capillaries. This tuft or mass of capillaries, it is termed as a glomerulus. It is termed as a glomerulus. So second component, that is glomerulus. Glomerulus is a mass of blood capillaries, and it leave the Bowman's capsule. As a efferent arteriole. Means blood enters into the Bowman's capsule through the afferent arteriole. When afferent arteriole enters into the Bowman's capsule, it forms the network of capillary. This network of capillary form the tuft or the mass or knot of the capillaries. अशी गुंडाळी होईल आणि ती गुंडाळी त्या कप शेप्ड कॅव्हिटी मध्ये अशी बेसेल की ज्याला आपण ग्लोमेरुलस म्हटलेला आहे. सो ग्लोमेरुलस इट इज द knot or tuft of 50 parallel capillaries. So near about 50 parallel capillaries from the tuft or mass, it is termed as a glomerulus. And this structure mainly involved in the most important process, that is the ultrafiltration. At the base of Bowman's capsule, we can observe the neck. This neck is lined by ciliated epithelium. So this is the structure of Malpighian body or Malpighian corpuscle. In theory exam, separate short note will be asked. Write a short note on Malpighian body. In NEET exam, objective may be created on this Malpighian corpuscle or Malpighian body. Remember that this is the difference between afferent and efferent arteriole. Afferent arteriole have bigger diameter than the efferent arteriole. हा point इथे महत्वाचा आहे, which does not observed in the juxtaglomerular nephron, because there is the no diameter difference. Here it is the cortical nephron. Now this is the Malpighian body or neck. Then concentrate on the next part, that is the proximal convoluted tubules. So second part, that is proximal convoluted tubules. In short, it is termed as a PCTs. So it is termed as a PCT, proximal convoluted tubules. These proximal convoluted tubules means convoluted portion of the renal tubule present at anterior side. So it is termed as a proximal convoluted tubule. These proximal convoluted tubules have average diameter, it is 30 to 40 micrometer is the average diameter of proximal convoluted tubule. Proximal convoluted tubules lined by cuboidal epithelium. So proximal convoluted tubules lined by cuboidal epithelium, but this cuboidal epithelium have the peculiarity at proximal free end have the finger-like projections. Remember this point. And these finger-like projections are termed as a microvilli. So presence of microvilli is the peculiarity of cuboidal epithelium of proximal convoluted tubule. We can observe the nucleus in the cytoplasm, but here it is presence of second peculiarity, there is presence of large number of mitochondria. Or abundant mitochondria observed in the cuboidal epithelium. So this is the mitochondria. So large number of mitochondria present in the cuboidal epithelium, This is the peculiarity. So proximal convoluted tubules means convoluted portion of renal tubule lies in anterior part of the renal tubule. So it is termed as a proximal convoluted tubule. These proximal convoluted tubules also lies in cortex region of kidney. Average diameter it is 30 to 40 micrometer and it is lined by cuboidal epithelium. But this cuboidal epithelium have the brush borders, that is microvilli and mitochondria. These microvilli perform the function, it increase the surface area for absorption, and mitochondria provide energy during active transport. Remember this point from NEET exam point of view, that is microvilli and mitochondria. And function performed by these proximal convoluted tubules involved, it is the main site of selective reabsorption. So it is the main site of selective reabsorption. Now third part, it is loop of Henle. This loop of Henle is made up by three components, that is descending limb, U-turn and ascending limb. So these are the three components, that is descending limb, U-turn and ascending limb. When you focus on this descending limb and ascending limb, first descending limb is thin walled, this is the difference, it is thin walled, while ascending limb is thick walled, Average diameter 10 to 15 micron is the average diameter of descending limb, where average diameter of ascending limb, it is 25 to 30 micrometer, it is the average diameter. This distal limb lined by cuboidal epithelium, while it is lined by flat epithelium. But the difference is that descending limb is permeable to water and impermeable to salts. This descending limb is permeable to water while impermeable to salt, while ascending limb is permeable to salts and impermeable to water, because this behavior or this mechanism helps in counter current system or amplifier, because this Henle's loop decide the concentration of the urine. युरिनचं कॉन्सन्ट्रेशन हायपरटोनिक की हायपोटोनिक हे ठरवणारा पार्ट म्हणजे हेनलेज लूप आहे, की ज्या हेनलेज लूपमध्ये हे बिहेवियर होणार आहे. वी आर डिस्कसिंग ड्युरिंग दी डिस्कशन ऑफ द काउंटर करंट सिस्टीम. बट लूप ऑफ हेनले इज मेड अप बाय डिसेंडिंग लिम्ब, यू टर्न एंड ascending limb. Descending limb is having the thinnest diameter of the renal tubule, 10 to 15 micrometer, where ascending limb have 25 25 to 30 micrometer. Descending limb is permeable to water, impermeable to salt, while ascending limb is permeable to salt, impermeable to water. This loop of Henle leads into the third part, that is distal convoluted tubule. Distal convoluted tubule, in short it is termed as a DCT, distal convoluted tubule. Average diameter here also 30 to 40 micrometer. It also lies in cortex region, while Henle's loop lies in medulla region. So distal convoluted tubule means the convolution portion of renal tubule present at posterior side, so it is termed as the distal convoluted tubules. These distal convoluted tubule have average diameter, it is 30 to 40 micrometer and lies in cortex region. So this distal convoluted tubules also lined by cuboidal epithelium, also lined by cuboidal epithelium. But the peculiarity of this cuboidal epithelium means this cuboidal epithelium does not have brush border, that is microvilli. So microvilli absent in this cuboidal epithelium. We can observe the microvilli, here we cannot observe the microvilli. Centrally placed nucleus. Second peculiarity, there is presence of few number of mitochondria. Compared to this cuboidal epithelium, there is a large number of mitochondria. Here it is few number of mitochondria. So here it is the difference between the cuboidal epithelium of DCT and cuboidal epithelium of PCT, and it involved in the function, that is site of secretion, site of selective secretion, and second, that is site of absorption under the control of hormone, that is ADH and aldosterone. Under the control of hormone, that is ADH, antidiuretic hormone, and aldosterone, means here absorption occurs, but this absorption occurs under the influence of ADH and aldosterone. It depends upon the condition, it depends upon the physiological condition, and it is site of secretion, site of renal secretion. So site of renal secretion. This is the distal convoluted tubule. Distal convoluted tubules end into the next part, it is termed as a terminal duct. Terminal duct, it is a small duct connect DCT, distal convoluted tubules, with common collecting duct, and it is the site where actual concentration of urine formed. Actual concentration of urine will be observed in the terminal duct, but the concentration of urine decided by the loop of Henle. So it is the terminal duct. So this is the posterior terminal duct which connect the distal convoluted tubules with the common collecting duct, and from this arises the part that is common collecting duct. This common collecting duct collect urine from different renal tubules, and this common collecting duct opens into duct of Bellini, opens into duct of Bellini. Duct of Bellini opens into renal papillae. Renal papillae opens into minor calyx. Minor calyx opens into major calyx. Major calyx opens into renal pelvis, and from renal pelvis it goes into the ureter, and from ureter it is to urinary bladder. So this is the sequence. So sequence of urine transferred from the common collecting duct to duct of Bellini, duct of Bellini to renal papillae, renal papillae to minor calyx, minor calyx to major calyx, major calyx to renal pelvis, renal pelvis to ureter, ureter to the urinary bladder. Okay, so this is the structure of nephron or uriniferous tubule. Once again, in brief, I will focus on these different parts of the uriniferous tubule. Uriniferous tubule or nephron is the structural and functional unit of the excretory system or kidney. Each kidney consists 1.2 million nephron. Out of this 1.2 million nephron, 80% nephrons are the cortical nephron, 20% nephrons are the juxtamedullary nephron. This is the cortical nephron. When we focus on the generalized structure, nephron is made up by the long renal tubule get associated with the glomerulus. This renal tubule blind at anterior side, opens at a posterior side, and shows different parts like anterior Bowman's capsule, neck, proximal convoluted tubules, loop of Henle, distal convoluted tubules, terminal duct which opens into common collecting duct. One part associated with Bowman's capsule is glomerulus, and glomerulus and Bowman's capsule form the Malpighian body. The inner layer of Bowman's capsule lined by specialized cells, it is termed as the podocytes, which have the foot processes and slits helpful in the ultrafiltration. Then proximal convoluted tubule and distal convoluted tubules present in cortex region. These proximal convoluted tubule and distal convoluted tubules lined by cuboidal epithelium, but proximal convoluted tubules cuboidal epithelium have microvilli and mitochondria which are absent, means microvilli absent in the cuboidal epithelium of DCT and few number of mitochondria present here. So both are involved in the selective reabsorption, but main selective reabsorption occurs in PCT, while selective reabsorption occurs under the influence of ADH and aldosterone in DCT, and DCT involves the site of secretion. Middle portion is the loop of Henle. Loop of Henle is made up by descending limb, U-turn and ascending limb. Difference between descending limb and ascending limb: thin walled, thick walled, average diameter 10 to 15 micron, average diameter 25 to 30 micron. It is lined by cuboidal epithelium, flat squamous epithelium. It is permeable to water, impermeable to salt. It is permeable to salt, impermeable to water, and helps in the counter current mechanism. It opens lastly into terminal duct. Terminal duct opens into common collecting duct. In this way, the urine is transported towards the urinary bladder. Thank you. From NEET exam point of view, we want to focus on this blood supply to the kidney because it is essential for the mechanism of urine formation. You know that from the heart, it is the four-chambered heart present in the human being, and from this left ventricle, arises the aorta termed as a systemic aorta. And this systemic aorta divides into several branches, and one branch of systemic aorta goes in posterior side. It is termed as a dorsal aorta. In abdominal region, this dorsal aorta bifurcate and innervates to the kidney. It is with the help of renal artery. Renal artery brings this oxygenated blood but impure blood into the kidney. When it enters into the kidney, it divides and forms the segmental artery. Segmental artery, when enters into the pyramidal region, it gets divided into lobular artery. This lobular artery into the pyramids innervate each and every nephron with the help of afferent arteriole. These afferent arteriole bifurcates and form the capillary, and the network of capillary is termed as a glomerulus. So glomerulus is termed as a tuft or knot of capillaries. This tuft or knot of capillaries, glomerulus, rejoins and forms the efferent arteriole. These efferent arteriole leave the Bowman's capsule and forms the network of capillary around the nephron, and this capillary network is termed as peritubular capillary network. This is peritubular capillary network, means each and every nephron is innervated by the network of capillary termed as a peritubular capillary network, means each part is innervated by the blood capillaries, and one branch of peritubular capillary network which runs parallel to loop of Henle, in juxtamedullary nephron. It is termed as a vasa recta. This is most important. It is termed as a vasa recta. So this limb of vasa recta, it is descending limb, while this limb of vasa recta, it is termed as ascending limb. So you know that this is the descending limb, ascending limb of renal tubule, but here it is descending limb and ascending limb. This means the opposite direction flow is essential for counter current mechanism. All capillaries rejoins and forms the venules rejoins and forms the renal vein, and this renal vein brings pure and deoxygenated blood released into the inferior vena cava. This inferior vena cava passes this blood towards the heart. That's why remember, this is the renal vein which release out the deoxygenated blood but pure blood which consists least amount of urea. असाही प्रश्न विचारला जातो, which of the following vessel consists the least amount of urea. सर्वात कमी असणारी ब्लड व्हेसल कोणती, तर लक्षात ठेवायची renal vein. पण चुकतो आपण कुठे, which of the vessel consists largest amount of urea असा जेव्हा क्वेश्चन येतो मुलं पटकेना लिहिता दॅट इज renal artery. पण हे पूर्ण चुकीचं आहे, कारण का युरिया तयार होतो by the cycle, that is ornithine cycle. It is occurs into the liver. So from liver arises the vein. It is termed as a hepatic vein. So remember, that is hepatic vein consists, that is largest amount of urea. So this is the most important point, that is hepatic vein consists largest amount of urea. प्रश्न असाही विचारला जाईल, make a correct sequence of the blood vessels innervates the kidney, dorsal aorta, renal artery, segmental artery, lobular artery, afferent arteriole, glomerulus, efferent arteriole, then peritubular capillary network to vasa recti to renal venules forms the renal vein, and renal vein release it into the inferior vena cava, and inferior vena cava brings this blood up to the heart. This innervation is essential because during we are discussing about the mechanism of urine formation, exact exchange of material takes place between the renal tubule and the capillary tubule, means capillary network. ही जी exchange होणार आहे ती दोघांच्यात होणार आहे, that is renal tubule. So this is the renal tubule, and secondly it is occurs between the peritubular capillaries. This is capillaries. So most important means some movement of ions occurs from renal tubule to blood capillaries, and some movement occurs from the capillary to renal tubule. So discussing the mechanism of urine formation, this point is most important, that is the material get enters and exit, means the movement occurs through the between renal tubule and blood capillaries. So it is about the blood supply to the kidney.